So, good evening everybody. Seems at least most people managed to find this room here. Not quite sure what's in the other room, but somehow I couldn't reserve it. So, where did we stop last time? We did RNA-seq data analysis. We looked through all this stuff and looked at our data and looked at our differentially expressed data. And maybe if it works, I quickly pull up this app that we had in the end. So I saved all the data that we had last time in one big in a file. No, that's the wrong one. Session set working directory to source file direction. Picture 1, texture 3, 6, 7, 8, RDA. Yes, that should be it. So what you can do in R is that you save all the data that you have currently running in all your variables in one big file. They usually have an extension RDA for our data file. And at the end of last lecture, I've done that so that we can continue where we've been before. So I say here load and load this RDA file, and now we see all the stuff of last time. And I should probably also load the libraries we had last time. We used my DEC2 library, and in the very end I showed you this RLC library that Sveta and I had developed in the last couple of months. And I only briefly demonstrated it, and I start this again because it's a good way to remind you all where we've stopped. Let's hope that it actually works today. Yep. One thing, right, we have a better projector today. Strangely enough, it only has an analog cable, but it seems to be able to display my whole screen. If it gets too small, you have to shout. So, you remember, who still remembers what we've done last time? Probably nobody. We had this we had this airway epithelial data where we had 4 different cell lines derived from airway epithelium from 4 different donors, and we wanted to know how this cell line reacts to dexamethasone. And here to the left, we see the result of our differential expression. On the x-axis, each dot is a gene. On the x-axis, we have the overall expression strength of the gene, simply averaged over all 8 samples. And on the y-axis, we have the log2 fold shapes. So remember, this is log2, so 1 means 2-fold, 4-fold, 8-fold, 16-fold up, and so on, and the same the opposite direction. 2-fold, 4-fold, 8-fold, 16-fold down. The color here is the judgment of this DEC algorithm, whether this is now a statistically significant change or not. Now, what does statistical significance mean? You can see here, if I click on any of these points, I see the corresponding gene. And here I can see that, yes, for this gene, in fact, the treated samples are always above their respective control sample. And there's a couple of cases with really strong differences and a couple of cases with quite weak differences. And of course, in this middle ground, we have lots of cases where the difference is not only not significant, but not even consistent. In some samples it goes up, in other samples it goes down. Now, what does statistical significance here actually means? It's probably pays to think a bit about this a moment. What we really want to know here is if now somebody else would redo the experiment using 4 different cell lines derived from 4 different donors, would they get the same result or not? Now, a result which has been confirmed in all 4 cell lines like this here. No, nothing happens. I want a good result like this here is more likely to get confirmed again in any of the in any further experience on new cell lines, while a result which isn't even consistent among these 4 cell lines probably won't be consistent either among the other 4 cell lines. This now is something which we probably will have to discuss a bit more in the next lecture. I want to talk a bit about experimental design because there's a non-trivial aspect in here. You might, if you think about your own lab practice, you might have noticed that usually if you do replicates, you would have one cell line and do several replications. So if people would have like here four treatment and 4 control samples, this might be 4 different HeLa cultures or 4 different HEC493 cultures. It's a rather unusual design. What we here have is that we used samples from 4 different donors. Now, if we imagine this here were a HeLa cell line and we saw this, and now the question is, if we read it with 3 more HeLa cell lines, would we get the same or not? This now depends on whether the kind of variation that I see here is a variation which is caused by simply doing the experiment multiple times, or whether it is caused by the different genetic backgrounds of these 4 different donors, who are after all different people from an outbred strain, namely humans. And that's quite important and something people keep forgetting when doing biology, that of course one cell line is only one representative, and saying it reproduces in the same cell line is not worth as much as saying it reproduces over many different cell lines, because only the latter gives you confidence that you have found something which of a general knowledge which really advances the general knowledge about biology and not the specific knowledge about the idiosyncrasies of HeLa. Which means whenever you hear the word replicate, you should be very careful. If it were for me, I would suggest to ban the term replicate from the biological terminology, because everybody means something different. Somebody says, I have 4 different replicates, and by this means they have prepared 1 culture, treated it once, and then made 4 aliquots for 4 sequence library. The next person means, I have my 1 culture, I have made 4 different passages, split this culture, and then did it 4 times. The third person said, okay, I used HeLa, but from 4 different labs which I collected to control for variation. And these people, they now use from 4 different donors. I would argue there is a certain clear, there's a clear quality difference, and the more care you take to make your replicates as different as possible, the more general will anything be that you find to agree over all of these 4 replicates, which is exactly the opposite of what your average novice biologist thinks replicates are. Most people who are for the first time in the lab think the important thing about replication is that you try to make exactly the same thing again. No, quite the opposite. Here you see the important thing is that you keep changing, like from one cell line to the next. But then we shouldn't call it replicates because we haven't changed it. We should call it independent samples. So I mentioned this now, but let's now go back to some technicalities. You can let this steep a bit, and then I guess we should have some discussion about what is a correct replicate or not, and when can you conclude what. And here in this case, where we have a so-called paired design, we will have to make a few more special considerations. Because we are not comparing, as you might have noticed, the average of a treated with the average of a control samples, but within each cell lines we check the fold change between them. Now, so I will now sort of have decided to make this from the didactics in some kind of sweeping back and forth experiments. We go through the whole test several times, always looking at different parts. I've unfortunately forgotten my note about what I wanted to start with, so I'll start with whatever comes first to my mind, which is probably what I've prepared here, and that was a bit about gene Z enrichment. You remember we had all these genes which are orange here. I can find them. We had this table res with the results. Let's make this console a bit larger, and you see here, this is what we did last time. We had our genes. We have here our p-values and our adjusted p-values. If I now take this and say arrange it by p-adjusted, library tidyverse, then we see those genes with the strongest adjusted p-values. They appear here on top, and they have really low p-values. Maybe sort of reasonably strong gene changes. We can quickly look at, for example, this top one. What's still missing here in my little automatic link chart thing is a possibility to enter a gene name here and find the proper gene. I would have to go around, but there's a little trick. Let's make another one of my little jumps, which I do now for lack of notes, with this code, which I only briefly showed you last time, where I said here you see how this works. The left plot, the right plot is described. You describe what the x and y axis is here, and then here the y axis is the counts of this gene, and how does it know which gene it should look like. Here this says we should look in the count matrix at the position gene. Now what is gene? If I say gene, then it tells us the number of the gene we've last used. Now I can also actually, if I remember right, I can also give it a gene name and not just a number, which would in that case be GPX3. And now the next time it reads this thing, it will read off the line corresponding to GPX3. And if I say update charts, subscripts out of bound. No, it complained because because my row names are not the gene names, but these things here, the ensemble. How do I get there? Where's P adjust? Here it is. So this is our top gene, the most strongly changing gene, which goes up here. Oh, it hasn't even shown us a number. So this is how we can use this, and now we want to learn a bit more about these genes. And there's two ways how we can go for this. One thing is that we say, okay, we have here thousands of genes, and probably only the top ones are the interesting one, and the rest is just incidental findings. The other way is we see all these genes, and they probably all have something in common. In practice, what most people like to do is the latter. They take all these long gene lists and they go into it with an idea of the more the better. If you get only 10 differentially expressed genes, you're disappointed. If you get 500 differentially expressed genes, you're happy. And if you write in your paper, we have found so and so many hundred differentially expressed genes. Whether this is useful, I don't know, but I thought, even though I have my doubts about it, I start with showing you the conventional way. First, and the conventional way is to do something which is called gene set enrichment analysis. I first show you how this looks like, and then we discuss a bit what it is and how it works and to which extent it makes sense and to which it doesn't. Now, gene set enrichment analysis, let's first start it. There is a package called Cluster Profiler, which makes these gene set enrichment dot plots. Once I show them, you will notice that you've probably seen many of them in papers, and now you know how to make them yourself with this Cluster Profiler package by you et al. Omics 16. By the way, you might have noticed that in the last write-up and in this lesson's write-up, I took a lot of care to always write the papers which describe each of the tools I use. There's a reason behind that. Something that we bioinformaticians always get annoyed about, and I think for good reason, is if people use our tools and don't cite us. The way how this typically happens is you have some big experiment and the big boss tasks the young PhD student to do the data analysis according to this standard workflow of the course, and then he follows this standard workflow and then tells the boss, yeah, I just did what everybody does. And of course he used in the term lots of different tools, but the young guy is too junior because it's his first year. He doesn't know about scientific etiquette and the importance of citing people, and the PI never realizes that actually bioinformatics is not pressing a button but doing a lot of parts, and you should at least figure out which buttons you've pressed and who to cite for this. So I now try to start to lead with good example and give for each, for everything I do, I have made here the citation and the link to the paper and so on. And if you now use any of this stuff, then of course you should do the same, especially if it's a paper by me, but also by the others. So what we now do is we use something called the gene ontology, and we will first see how it looks and then talk a bit more about what it does. And we use this organizational database package, which means organism Homo sapiens entrée genes database. This is Bioconductor's way of making what we call annotation packages, which just collect all the information which is known about the genes of Homo sapiens according to the entrée gene database. Entrée genes, as you might know, are those gene names which are just numbers. You sometimes have gene names which start with E and SG something, like these things here. These are the ensemble ones, and entree are those which don't have any gene names, only numbers. Now, let me first here run this code and show you what it produces, and then we discuss about what it does. I think it takes a couple of minutes. While it calculates, I can already explain a bit. What gene ontology is two things. First, it's an ontology, and I'll explain you in a moment what an ontology is. And second, it's a database. It is a means to write in a digital readable form everything we know already about what gene does what. So if we know that a gene is, say, a methyltransferase, then there is a GO term for it. And of course, it's important if you just had a database which writes gene X is a methyltransferase, the next person writes gene Y is a DNA methyltransferase, and the next one writes gene Z is an enzyme which transfers methyl groups, when you have no way of figuring out that these are actually three different things that do the same. What we need is something that computer scientists call a controlled vocabulary. We make a fixed list of terms, and this term says the word is DNA methyltransferase. It is not enzyme to transfer methyl. And everybody is bound to use these words and only these. This is what we call a controlled vocabulary, and it's a very important thing. And of course, it's a big work to decide which words to use, which have exactly the right granularity, that they are not too fine to distinguish things, to make it too burdensome to annotate things, but also not too broad that it doesn't make much sense. Once you have these terms, you can bring them into connections. You can, for example, say there's enzymes which transfer chemical groups in which subdivide into methyltransferases, acetyltransferases, whatever transferases. The methyltransferases go into those which have DNA as a substrate and those which have RNA into substrate. And you see all these controlled terms, they come into something, they have a hierarchical regulation where one is a subcategory of the other one. And the gene ontology now has these two parts. There's first the gene ontology consortium, which comes up with all these terms and draws all these arrows, which they labels with is a or is part of. And the other thing is when the teams which are responsible for the individual organism, and then, for example, take each human gene or each Drosophila gene and annotate and make the connections between the genes of an organism and the GO categories. And we now here use both, and now we run this, and now we do an enrichment analysis. What I've done is I took all the list of the genes which are upregulated, so all the orange dots here on the upper half, and ask, are there certain categories enriched in them? By enriched, I mean, let's say 2% of all the genes in the human genome are annotated to be a methyltransferase, but among these orange ones, it's not 2%, but 5%. Then we call this an enrichment and say, obviously, the process we are studying has some connection with methyl transfer. Now, I always keep saying methyltransferase because somebody was bothering me with some DNA 450K array data, so this is in my mind, but it actually has nothing to do with our data. So let's see what we have in here. We have this EgoCC, this object that this cluster profiler, it makes some analysis, and it gives us some result in this lengthy thing, and then it comes with a couple of different ways to plot it. For example, this fancy dot plot thing. And that's the ones which I said you might have seen in papers before. This is what we call a gene cluster profiler dot plot. So how does this work? Each of these things is one of these GO terms: anchoring junction, adherence junction, actin cytoskeleton, and so on. The x-axis tells us the ratio. So basically, the thing has found that how does this now work? That 12% of our genes, of our orange dots here, 12% of these orange dots are annotated to be part of a cellular component anchoring junction, and this seems to be much more than we have in theory, than we have in the whole genome, and hence this is considered to be highly significant. So the p-value here shows us how significant is, how much stronger this gene ratio is is compared to the proportion of this kind of genes in the general list of all human genes. And this circle size tells us how many genes were in there. So in total, I think these were 300 orange genes, and here, according to these, something like 70 of them are associated to anchoring junction or adherence junction. A few more to actin cytoskeleton. Now we are getting to stuff like cell substrate junction, which we can already guess is probably just a supercategory which encloses anchoring junction a bit more. This is why we have another plotting function which is called geoplot, and it shows us something which one calls a DAG. A DAG, D-A-G, that's a directed acyclic graph. If you squint a bit, you will notice that you will first see some kind of tree structure. So here you see the root of a tree, cellular component, and here with the root sprouts different branches, the different cellular components which are described in GO, namely an organelle, a extracellular region, the cell itself, the junctions between cells, the ECM. The organelles again are divided into membrane-bound and non-membrane-bound, and so on. So we can imagine how GO has this kind of tree structure where you have a root. Those people who haven't been confronted with computer scientists might not know that, but in computer science, trees grow downwards. The root of a tree is always on top, and it branches downwards. In each level, you have a branch which then sprouts more branches. And we go this, and here we have all these nodes, but this is a bit more because sometimes we have connections between branches, and that's called a DAG, a directed acyclic graph. Directed because there's always a direction. This is part of that, or this is a subtype of that, and not the other way around. Acyclic because all these arrows never point in a big circle, which wouldn't make sense, because then we would have one thing is a part of the next, and next is part of the third one, this is again part of the first one. That can't be. So this is what this geoplot does. It takes out of a database all this knowledge about how the GO terms are connected and brings them all in one big shape. And now we can study them. And we can now already see what we already thought, that something like the anchoring junction and adherence junction, that wasn't actually two different findings, but it's simply more or less synonyms, because if we are enriching for one, we are also enriching for the other one. The GO is specific for this thing, that it has this DAG structure. We will see in a moment a couple of other collections of gene categories which don't have any relations between them, and you just have to check for overlap. And this is what we have here with a third plot, the IMAP plot. And
I have a question.
Yeah.
So the adjusted p-value, is it just something that's grabbed out of the DSIG example?
No.
Or is it calculated for
It's
this
calculated here, and I come in a moment to what actually this p-value means. Here now you see everything which came out of it. Somehow there's a few more than we saw before, and we see connections if there's a strong overlap. The list of all the genes which are annotated to, let's say, cell substrate adherence junction has a strong overlap with anchoring junction. So probably if you're enriched for one, you're enriched for the others. Now, there's actually not one geontology, but there's three of them, called cellular components, CC, molecular function, MF, and biological process, BP. You can see these are simply three different philosophies how you can describe your biological world. You can either ask where in the cell is it, that's cellular component. You can ask what does it do in the cell, that's molecular function, or what does it do in the whole organism, that's biological process. Here I've now asked my enrich object to enrich for CC. Now here you can see how this whole thing actually worked. I used my result thing. You remember this was the result table from DESeq. I filter down on only those which are significant, so which have a significant adjusted p-value, and a fold change greater than 0, so which are going upregulated. And now I get them when I use the pull command to get a single line, a column, namely here the ensemble IDs, like this. Now I have all the ensemble IDs. And now I hand this to the enrich go function. The enrich go function wants a list of ensemble IDs, and then it wants a few more things. It wants a database which tells it how to annotate gene ontology to genes, and here it takes this org-hsegdb. If you go onto the bioconductor webpage, you will find a whole list of more such objects, not only for Homo sapiens, but also for all the other model organisms. It wants to tell us what we've actually given them, how it should interpret these things, what kind of key it is, and there's somewhere a list of different key types that you can give, and ours is ensemble IDs. It wants to know which ontology. If I now switch, for example, from cellular components to molecular function, we can see this, what it does. And now I again have to wait for a few seconds to see what this does. And then there was the last thing, which is called the universe. The universe is the following. The universe is unfortunately not as fancy as it sounds. I always said we compare the fraction of this, the fraction of the GO category among our upregulated genes with a fraction among all genes. But what do we mean by all genes? If we would take all genes which are annotated on ensemble, basically everything is enriched, because ensemble is full of genes which are never expressed and don't and hence will never appear in our GO thing, and we don't know anything about it. So it's important to tell it which genes we've actually been looking at, and not simply say it compared to all genes. And this is what's called the universe. All the genes for which we could in principle have noticed that they are upregulated. And if you look at this, it seems that we can notice upregulating only of all the genes which have an average expression of at least that much, of at least 3-4 counts per gene, or maybe a few more. So that's what I then use for the universe, that I say all the genes which have a mean above 5 or so are what should be the baseline comparison. Or I think here I now set all the genes which have not an NA in the adjusted p-value, which brings us back to what these NAs mean, which I think I wanted to explain later. So, now what does this whole thing mean? It sort of makes sense. We are looking at epithelial cells. We are looking at dexamethasone, which is a glucocorticoid. We are trying to do asthma, but on the other hand, if I have an asthma attack or if you have any bronchitis, I didn't know that my epithelium actually comes off. So I've Googled a bit for it, and I've learned, and maybe those of you who worked a bit more with these things will have known that dexamethasone and other glucocorticoids are known to not only suppress the immune system, but also to suppress the endothelial to mesenchymal transition, and that hence, if these cells, because hence they will influence genes which are usually associated with EMT, and these again are of course genes which are associated with cell-cell junctions. With a bit the catch with these things, whenever you see something after some Googling, you can come up with a story of why this makes sense. Whether this really helps us to understand now what dexamethasone does, who knows? Just to stay with this theme here, I show you one other aspect. Instead of a gene ontology, what you can also use is something called the molecular signatures database, which is done by the Broad Institute. And they have here this list of gene set collections, each of them a long collection. And now I take one collection, here the hallmark data set, which has all browse 50 gene sets. And here see which gene sets we have. Here's, for example, one hallmarks of hypoxia. I like that one because it's just a curation of many different experiments and finding a couple of core genes which always seem to be upregulated in all experiments where somebody tried with hypoxia. To do this, of course, we now have to use, we have to download here the GMT from here and go through it. And I think I will now not go through these parts. You can study this yourself when I've uploaded it. But you see the general gist is the same. Yes, I used it here for my, but I quickly run it. So I've downloaded here this file which has all the, which has all these hallmark marks. You have the hallmark TNFA signaling and the corresponding genes, and a bit lower in the table you will get here on the left other terms and so on. Then I give it again now to a slightly different enricher function and check the dot plot this time, and you see our main thing is again what I've already mentioned, hallmark EMT, epithelial to mesenchymal transition, and now you wanted to know what these p-values mean. So let's try to do what we've just done now manually. So why do we see EMT upregulators? Now what we have here is, what we used here is this list here, hallmark EMT, and we have here the list of the genes which are somewhere here, I think. Whoop. If I open this, here's all the genes which are known to change in EMT. And if I now do this, and if I now want to do manually what this complicated enricher profile has done, I can do this here. You see, I take my result set again. Where did I do it? Yes, EMT genes was when I first wanted. Here I take this GMT thing, which you remember are all these things. Now I filter out those which have EMT. Now I see the gene of EMT. I pull the gene column. I save this in this variable. So this is now the variable with all those genes here from the EMT list. What now gene set enrichment analysis means is that you do the following. We take our genes, we take again, we filter this down to our universe, all those which we could actually test. Now I add here these two, could not find function res. Why there is no function res? Here is result. So now here we have our result table, and now I've added here to the right two new columns. One column whether it was significantly upregulated, so whether it's orange, and one column whether it was in this list of EMT genes. And what I can now do next is that I now simply group by these two things. So I sort my rows into 4 groups where both are false, one is true, and so on. And I tally them, and now I get this thing. So we have 12,000 genes which are neither upregulated nor an EMT gene, 144 which are EMT genes and not upregulated, 761 the other way around, and so on. This is maybe easier to see if I write it in this kind of table. And now we can see what enrichment means. We can ask what is the proportion of significantly upregulated genes among the non-EMT and among the EMT genes. And you see it's much higher among the EMT genes. You might now remember, if you remember back to the very beginning of the course, I told you about the lady tasting tea, where we made this kind of contingency table. The columns were the lady says that the milk was put in first, or the tea was put in first, and the rows were milk or tea was actually put in tea first. And we asked, is there some relation between them? And we used the Fisher test. And we can do exactly the same by taking now here this thing and doing a Fisher test. Now it calculates the Fisher test and tells us, yes, these two objects can't be independent. We get a very low p-value, and this is the p-value we see, the p-value for this being enriched. Now, we have, typically, you might remember in the lady tasting tea thing, the crucial point to get significance was to use enough cups of tea. The more cups we use, the smaller the p-value. Back then we calculated that if you use less than 8 cups, there's no way for the lady to prove her talent. If you use 20 or 30, the p-values can get lower and lower. Here is the point, the more genes we have in our gene set, the lower our p-values will get. So these enrichment p-values have a curious property. In order to get good p-values, we don't need many replicates, but we need big gene sets. And this shows you that there's something fishy about them. We are not, our unit of replication is suddenly not a sample, but it's a gene. And there's been a lot of debate in the field about whether this makes any sense at all or not, but I think the consensus is simply never take these enrichment tests at face value. They are sort of just a hint where you go, and there's really no way of saying when something becomes significant. Probably it's never significant, but it may make sense or not. Now, what was the next thing? Shrinkage, exactly. If we now go back here to this thing, you might have noticed already that we have this strange curved decision line. I always feel reminded of a trumpet. Like imagine this blue thing is my trumpet, I blow in from the left, and the horn, I blow in from the right, and the horn is to the left. Why do we have this? The reason is, if you look at the counts axis at the bottom, the counts here are really low. These are genes which have an average of 1 read per sample. Here this is 10 reads per sample, which is really not much. So if we just happen to have 1 read in all the control samples and 3 reads in all the treated samples, when we seem to have a 3-fold upregulation, and if it's 4 reads, then we get a much higher upregulation. With thousands of genes, it's quite likely that there's quite a couple of genes where really we just arbitrarily have dozens of reads extra on one side and not on the other side. And what DEC does is that it actually calculates this, and we'll see a moment later how it calculates that, and hence it accounts for this and makes this curved decision boundary. So in order to show you how this might happen, I quickly do this demonstration here. I take let's imagine we have, I used to have a slide which I've now forgot to include. What we have to look at here is something called Poisson noise. And whenever you work with sequencing, it's absolutely important that you understand Poisson noise. To understand Poisson noise, what you have to imagine is the following. We start with a simple example. Imagine I would have here a big brown bag which is filled with balls. And these balls are white and red. Actually, 10% of the balls are red. So my bag contains 1,000 red balls and 10,000 white balls. This is my bag. And now you don't know how many, which percentage of a bag is red. But what I do is I allow each of you to take 10 balls from this bag without looking and look at them. And now one of you takes 10 balls without looking. You might remember the sample command, which always takes something and just takes a sample out of 10. And here you see what the first gun does. He gets 1 of the 10 balls are red. The next one also has 1, which is of course what we expect. Here we have 0. Here we have 1 and so on. Now let's always ask how many of them are red. Red. Some. 2, 1, 0, 2, 2, 0, and so on. I can use, instead of taking, I can divide by 10. So I take the mean when we see now our estimated proportion. This guy says there's no red balls. Here's somebody who says 20% red balls, 0, 10%, and so on. If I now would do this, replicate this now 1,000 times, and I draw a histogram of this, then you now see what we get. We get a lot of different opinions. There's people who say it's 0, 1, 2, and so on. If I now allow each one to not draw 10 balls, but 100 balls, things look much better. See, 10 of them red, 12, 11. If I take my mean, it's 9%, 9%, 8%, 12%, 10%, 9%. This looks quite good. And if I would allow him even to take 1,000 balls, then it gets even closer to the proper result. Now let's do here the replication with 100 balls and draw a histogram. And now you see here how this histogram looks like. You have this characteristic shape, and this is called a Poisson distribution, because some guy called Simeon Poisson looked at this first in 1870 or so. The Poisson distribution has an important property. The mean, the average, which we would get after many of these replicates, is always pretty much exactly at the expected value, 10%. And the spread of this thing is the square root of this. So maybe I shouldn't use a mean, but I should have used a sum here. So that you can see it's the average is 10, but actually it's a bit less than 10. It's maybe because I've done a slight mistake. When you take 100 balls out of my bag with only 10,000 balls, where it's not enough. Once you take the first ball, you have a probability of 10%. The next ball has a slightly lower probability. Every time you have already taken a few balls, the probability goes down. This is called the binomial distribution. What I'm imagining is the Poisson distribution, where we imagine that the bag is so big that it really doesn't change the fraction if you take one ball out. And in that case, Poisson's rule says that the expectation is, of course, to get what we expect. 10 of 100 balls will be red. And the standard deviation of this expectation is the square root of 10. So we should get something between 10 minus square root of 10, so between 7 and 13. Should be one standard deviation. So 68% of the values should be between 7 and 13. 99% of the things should be within two standard deviations, so between 4 and 16, which is more or less what you see here. What does this all have to do with sequencing? Well, if we take a sequencing library and we amplify it, when we have quadrillions of molecules in our fully prepared and amplified sequencing library, which we then put onto the flow cell of a sequencer, and when we sequence maybe 10 million reads. These are the 10 million, these are the balls we draw out of a bag. So our bag, let's say we want to quantify some gene, and with this gene we have seen 5 counts. To transfer to my bag analogy, the whole bag, this is the quadrillionly many molecules which have been in the prepared library. The balls which you pull out, that are the 100 million reads that we actually get back from the sequencing. And the 3 balls from the gene at interest, these are the red balls. So we want to quantify a specific gene, and so we imagine that all the reads from this gene are painted red. And if we now see a gene which has, if we now see for a gene that we got 5 reads from this gene, then we can imagine if we just had sequenced a second aliquot of the same prepared library again, it wouldn't have to be 5 again. So we can make this experiment by saying, I take here a random value with mean Poisson, R-Pos 1,5. And this would now be the following experiment. I have one prepared library, and I take many aliquots and put each of them on a different lane of my flow cell. And then even though I know that the concentration of this gene is exactly the same in each flow cell lane, because after all I took aliquots and I have mixed well and shaken well before I did it, still I sometimes get 3 reads, sometimes I get 5 reads, and so on. If now the average number of reads is not 5 but 500, when we are much closer to the real value. Okay, on an absolute scale, we are now not off by only 1 or 2 reads, but we are off by 20 or 30. But on a relative scale, it's only a few percent that we are off, and not 30, 50%. And the rule of thumb, how much we are off here in my 500 case is, so let's take not 1 at a go, but take 100 at a time, and I make my histogram here. And here you see how this looks like. Let's take enough of them. This is how it now looks like. You see it's more or less normally distributed. The expectation is 500, and the width is square root of 500. What's the square root of 500? It's 22. So it's something like from 68% should be within 22 counts and 95% within 44 counts of the expectation. Which means, now what is 22 over square root of 500? So this 22 over 500. This is of course the same as 1 over square root of this. And this is the precision. 68% of the data, so one standard deviation of the data, will be within 4% of the true value. And most of the data will be within 8% of the true value. So as general rule of thumb, if you have a count of 500, you can say this gives you the quantity, the concentration in your library with a precision of 4%. But if you have only 5 counts, you get a precision of 44%. You can be really way off. And this is one standard deviation. You know, you can easily be 2 or 3 or 4 standard deviations off. So whenever we count things, we have to be acutely aware of this thing, that a count cannot be any more precise than this. And this, of course, not only applies to reads in the sequencing, this also applies to cells when you count with a counting grid in the microscope, with accounts of flies with red and black eyes, or to whatever experiments you want to do. Whenever your experiment includes counting, you have to be aware of this Poisson noise. And it means that all the many genes in a typical RNA-seq experiment, for which we have less than a few hundred counts, we really don't get them very precise. We can only see them with a precision of 10-15%, which, on the other hand, if we are looking for strong changes of a gene, we might get way over. This might be good enough with precision. If we hope to see changes like that the gene goes 3-fold or 5-fold up, then knowing the precision only by a few, getting the precision wrong by a bit doesn't matter that much. But it also explains why we have so many genes here which seem to be much stronger fold changes. But this gene is here up 50-fold up. What is it? 2 to the 7, this is 128. So this seems to be 200-fold up. Is it really 200-fold up? Who knows? It might be that we are just running for this imprecision, which means if we now want to know which genes really affect strongly to dexamethasone, it might well be that these small genes, these genes here, have a much stronger response than these genes. And if we repeated this experiment, we would see in the repeat that this gene suddenly sits down there, while these genes, they stay as they are. To see this, we would simply calculate the standard error of this gene. You might have noticed that the results column actually has this information. Each fold change here has a standard error, and sometimes it's quite big, the standard error. Here, for example, the standard error is larger than the fold change, which is why it doesn't have a significant p-value. Now, what we want to do is we want to be able to tell which genes are really the strongest. And for this now, what we expect is, that's a nice board, no. If we now would have here log fold change, and here it's 0, so this is up, this is down, and now we have 2-fold, 4-fold, 8-fold up, water, and we have now one gene which sits here at this value, and another gene sits at this value. But this gene, because it has such a low expression, has a really wide uncertainty that basically all these values are plausible, while this gene here, let's put it here, has a really precise estimate, then maybe we better go with that gene, because if on average we know that typically in such an experiment it's rare to have a fold change larger than 5, so that most fold changes sit in this range, then we might say, given that this is the range that we expect, and these are all the ranges that are comparable to the data, maybe it would make sense to assume that this value should actually better be here, somewhere in the green area. While in this thing here, let me draw it somewhere here, while that one here you might say, okay, it sits here, it's probably more on the left side than on the right side. It can't be here because we know it's somewhere in this area, and there's that one may be more probable. So this is something which is called empirical Bayesian reasoning, which is a big word for just saying you first look over all your genes to see which kind of fold changes actually happen. And if we assume that these are all exaggerated here, then we can say actual fold changes sit here in sort of this range, and they never go much beyond that. And if now a gene is up here, we should only believe it if it really has a low uncertainty. But if the range of plausible value given by its standard error reach all the way down there, we should probably take rather these values. So we now replace each of these genes by a value which is closer to 0, and we move it, the more we move it towards this range of plausible values, which are seen here. And we do with such that we move a gene strongly towards 0 if its fold change is very uncertain, and weakly towards 0 only a bit if we are quite sure that it's already at the right place. And that's what we do here with this function, which is called log fold change shrinkage. And what log fold change shrinkage does, this is a rather new idea that you should do that. We tried to promote this in DESeq2 when we published the paper a couple of years ago, but it didn't quite take off, partly because our original shrinkage method didn't quite work well. So Mike Glove, who was back then the PhD student who actually brought forward the DESeq project, and is now has now his own group. He worked at it and came up with a better idea, and that now works really quite well. So here you see now the shrunken fold changes, and I show you how this now looks like. Whoop, 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 whoop. Here, now you see it looks very different than before. This trumpet shape is much smaller. There is still some trumpet shape, but now it's real. Of course, the decision boundaries. And now these genes, they have all the genes here have been moved down quite close to that. There's nothing left here of the weak ones, and of those there's much fewer of them here. This is now quite interesting because instead of saying all these 1,000 genes, all these 300 genes are all differentially expressed, we can now look at a few of these genes and say these are interesting. These 4 or 5 genes up there, they react way stronger than all the bulk down there. So maybe it's more interesting to look at these here and try to reason, instead of doing GO enrichment and so on, it might be better to just look at these specific top 10 genes on this list and say these are maybe the genes which really tell us what's happening to these cells, and these genes down there. There's one important thing. So I've been working on this topic of differential expression analysis quite a bit, and to be honest, the more years I've been working in this topic, the more I had the feeling that actually there is a mistake in the very beginning of this whole enterprise. Everybody does RNA-seq experiments. Whenever you have some kind of new system or question and so on, first thing that people do, let's do an RNA-seq. That's cheap and easy, and you get a list of genes, and then you can think about what to do with that. But and people always ask how many genes are differentially expressed. And now, an important point to realize is this question is fundamentally flawed. It's actually stupid, and I'll tell you why. If you put dexamethasone on these cells, we know that some genes will change. After all, it's a glucocorticoid. Furthermore, we know that whenever you change a gene, this changes other genes. Especially something, a messenger like a glucocorticoid or hormone will change some transcription factors, which will change a whole bunch of hundreds of downstream targets. These will contain transcription factors among them, which will change another 100 genes. These will contain among them maybe some allosteric regulators, which then switch the state or phosphorylate something else, which again changes the state of a cell. In the end, it is completely impossible that a gene doesn't notice somehow that it changes. So if you do this, if you simply, usually the null hypothesis is stated as the gene is not affected by the treatment. So if you ask which genes actually change expression due to the dexamethasone treatment, you don't have to do an experiment. I can tell you all of them, every single one. So why do we do it? What is it we really want to know? Well, if we have a gene like this, and we now come to a conclusion that these are plausible values how the gene changes, with 0 just smack in the middle, by the biological logic we can say this gene obviously has to change. It can't be that it's completely decoupled, but we can't even say in which direction. So what significance in RNA-seq really means is not the gene is differentially regulated, but rather the signal of this gene is strong enough that we can say with some confidence that we got the direction right. Which of course shows you why it's pointless when people make these papers in which they write, we have found 500 genes to be differentially expressed. The correct formulation would be, we assume obviously all genes will somehow react for the treatment, but for 500 of the genes we can tell you roughly which direction it's, whether they went up or down. It's of course very different because when people then say this treatment must be stronger because it caused more genes to be differentially expressed, the real thing is the data was better, so we can tell the right direction for more of them. This also tells you now what this 10% false discovery rate means. It means we expect that for 10% of them we get the sign wrong. Also, it shows you why we have this problem that I said we can't really say whether the genes which are far out of the MA plot are really the strongest, because all that the p-value tells us is that we can confidently say that we are on the right side of a 0. It doesn't tell us anything on whether we are right about how far out from the 0 are. This is the value of his work by Mike here, that now we find some kind of ranking which tries to pull the genes towards 0, for which the data is too bad to really clearly say how far out they are. This is something which we call a variance bias trade-off, because now we are biased. We have told our method to tell us a too small change if in doubt. So we now know that most of these fold changes are an underreporting, and what we now see out there are not necessarily, at least those genes for which we can confidently say that they are far out, but there might be many more genes, especially here in this region, which are also far out. So I mentioned this in some detail because this is really one of these things where it pays to think hard about how to talk about different. What your experiment really does in order to be able to understand what you can do with your data. Now you also see a bit the flaw of a gene set enrichment analysis that we've done before. We used this arbitrary cutoff and said, tell us what all these orange genes are enriched in, and these orange genes are those for which we know the correct direction of change, not those which reacted to a treatment. Hence, it might be much better to now do our gene set enrichment analysis with these ones here, and maybe it makes an arbitrary cut and say, let's take the top 50 of them and see what they are enriched with. I wanted to try this before the lecture and didn't quite go through it. I tried it quickly and noticed you get somehow different results, but I leave it for you as a homework if you want to try. So, what was the... let me quickly check my notes. Yeah, homework set. Yes, that was the shrinkage and so on. Questions on these things? Yeah.
If you could go back to the plot, please. I was a bit confused. Maybe it's also because it's late. There's 3 bluish dots in the upper half now.
Yeah, I also noticed them. What's happening here?
Yeah.
There is some disagreement between what the test does and what because the shrinkage and the significance tests are now calculated with 2 different algorithms. And you see they somehow agree by saying, okay, if we can't even say if it's not significant, which means that we can't say which direction it goes, it's best to simply report 0. Not because we think it's 0, but because that's the safest thing to say if you can't say anything. But where you should give up and simply report 0, the 2 functions, the shrinkage function, the test function, seem to have a slight disagreement. I would say this is an imperfection of Mike's method. Let's see whether I come up with some way. It also reminds you that maybe when I said this is probably a very interesting gene, maybe it should be 0, so it's still not really perfect. I feel a bit uneasy about those. And if I now really wanted to choose which one to follow up, if I have only money to follow up one, I would probably better take that one than this one, because that one seems to be well inside the area. But yeah, the whole thing is still far from perfect. And that's now talking about something as simple as bulk RNA-seq comparing, seeing the effect of simple drug treatment. We can't quite figure out how to analyze it best. And now everybody goes for single cell. So we biostatisticians won't get out of work soon because things we do now are still not quite perfect. Other questions? One last thing, I quickly, we started a few minutes late, is people asked me before about normalization and how that works. And normalization is something people always feel uneasy about. So let's look at this quickly by taking our counts object here, our counts matrix. I still have it here, counts matrix, and I simply take the first sample, so the first column. And I take the second sample. And if we want to compare the two, we should see how they look if I plot them against each other. This is now first column of the counts matrix plotted against the second column. And now I should probably say comma aspect ratio is 1, so that this is sort of on a square. And you see this is how it looks like. I can make an AB line, so a line with 45 degrees to see whether this is already comparable. And as you can see, it seems that the first sample is sequenced a little bit deeper than the second sample. If we now multiplied all these values from the first sample with a value which is slightly below 1, maybe we multiply this by 0.9, then maybe they would sort of center around this line. One way to see this easy is to look for each gene at the ratio of the first to the second sample. You see this is a ratio of the first to the second sample. I put this on the y-axis, and on the x-axis I put simply their average. Let me make this like this here, plus. So as you can now see, if you can read this, the x-axis is now the x-axis is now the average of the gene over the two samples, and the y-axis is the ratio. Looks strange here, the ratio goes way more up when it goes down, obviously, because if a gene is higher in the one, it can go up arbitrarily, otherwise it can go down only to 1. So you might remember what I've always said, whenever you plot a ratio, you have to plot it logarithmically. So I tell it I want the y-axis logarithmic. I'll also make the x-axis logarithmic, because you see most of the genes have, most of the plotting space is now used by these few really strongly expressed genes, and now it looks like this. This is how a typical, this looks a bit like the MA plots that we've already seen, but now it's something very simple. On the x-axis the average, just averaging over two samples. On the y-axis the the ratio between the two. I now make a horizontal line at 1 to 1, which is here, and we've just said this might be, let me make here something like call gleich adjust color black, comma alpha, which means slightly transparent, half transparent. Where's my x logarithm like this? Now you can see this. Now you can see a little bit better. Maybe if I do it like this, where these things plot on top of each other, and what you can now also see if I now make here my line, that the line seems to be more or less in the right thing, but maybe a little bit too low. Now, one way how to see this a bit better is that I make a histogram only of the y values, because here it's really hard to see how many points are plotted on top of each other. So let me take just here my y-axis, this side, this part here, where I divide 1 by the second. I take the logarithm because it's a ratio, and now I plot a histogram of them. Maybe I make a few more bins, 100 bins. That's it, 30. 60. And again, I make my AB line in here in orange. Sorry, I have to put it at 0. And you see it sort of hits the middle, but not quite. One thing which I could now take, I take all these ratios and I ask what's the median. And comma NARM is true. It's 0. It's exactly 0. That's a bit disappointing because usually it's never 0. Let me show you here the histogram for the first against the third column, and there the line is here. B is at 0. And if I now go a little bit to the left, then it goes through the median. And this is now what I do. I just divide all the columns of this third sample by this minus 2 to the minus 0.12, and I get this. What's the logic behind it? First, scaling. We assume that in RNA-seq, this problem of normalization solely stems from the fact that sequencing depth is arbitrary. How many reads you get out of your RNA-seq data in the end is, it doesn't really, it only depends on how you load the flow cell. If you take a little bit too little material, you get less reads. If you take too much material, you also get less reads because your clusters overlap. And this can, of course, differ from lane to lane. So it has no biological meaning whatsoever. So what you need is a scaling factor and say all the values from this sample should be multiplied by 0.8 in order to make it compare with this other sample. And all that we want is that we just find a set of scaling factors. We call them size factors in DE-seq, with which we normalize each sample so that afterwards, if we plot them against each other like this, like this, the orange line goes, the 0 line goes straight through the middle. And how do we find these values? Actually, in the most cases, it really works as simple as I've just shown you with just taking the median. It can work a little bit better by kicking out all these lowly expressed genes before taking the median. This is probably why I just got my exact 0, because I fell on one of these. But in the end, the automatic size factor estimation of DE-seq does exactly this. It cuts off a little bit at the bottom when it compares each sample. Actually, if I would compare each sample with every other sample, I would get, of course, one size factor for each pair. So if you have 10 samples, you might all know the old math, high school math question of if 10 people shake hands, how many hands are shaken? And you have to say, well, it's 45. And here, if I do what I've just said, and I have 10 samples, then I will get 45 size factors, one from each comparison or each handshake. That's bad. I only want 10. So I try to find a compromise, and the way how we do this is simply we compare everything to just one specific reference sample. And because we are worried that this specific reference sample then might be somehow treated, might be different to all the others, and might not be on equal footing to all the others, and this might introduce some batch effects. We don't take an actual sample as reference sample, but rather an imagined sample, and as imagined sample, we simply take the average through all of them. So for each gene, we take the average over all the samples. When we came up with this imagined reference sample, which for each gene simply has the average expression over all the samples, and when we try to make every actual sample comparable to this imagined average sample by just taking the median and making it on 1-to-1. And that's all we do, nothing more. There's even one more thing we don't do. We don't actually divide. We just take the number and store it. And it's important to take the number and store it and not actually divide, because as we've learned earlier today, the Poisson noise depends on how many reads you actually see. If you see 10 reads, you're more precise when you see 5 reads. And this is the actual reads. This is not the reads after division by the size factor, but before. Because if you sequence deeper, then of course you're really more precise. So in order to get optimal power, we have to keep the information about how many reads it really was, and how each read has to be weighted, and we have to process them separately through the two things. And then this is why we don't use the ordinary t-test, because the ordinary t-test only wants one set of numbers, but we want something called a generalized linear model of a negative binomial family, which is just a fancy way to accomplish two things. First, we are able to come in with the size factors independent of the counts. And second, this GLM model will take the counts to calculate the strength of the Poisson noise, and will take the size factors to then weight it correctly, and then it will use something called a dispersion estimation, which we also still have to discuss, which is just within group variance, and then it comes up with a p-value in that manner. So, by this, we are finished for today, I think. So, thanks all. Has everybody filled out the attendance list? If not, please do. Oh, I was wrong. I thought that the room was taken next week, and this is why I said next week there's none, and it's today, but it was wrong, as you've noticed, which is why we are here. So next week the room is open, so we can in principle use it. But I don't want to promise yet that I'll manage to prepare things till next week, so I'll tell you on Monday whether it's happening. Mir doch gerade was. Ich habe ja nie hier dieses Dings hier abgebraucht.